Good afternoon, viewers and attendees. How is everyone doing today? I hope everyone is as thrilled as I am for today's webinar. Our previous webinar last April 25 tackled Bioinformatics for the betterment of the Philippines. For today, we will be learning about the opportunities and challenges of bioinformatics here in the Philippines. I'm Diane Changko.
And I'm Elijah Rivera, and we are now officially starting the webinar on advanced bioinformatics for research purposes. SIHAY NBO is a national inter-high school biology congress organized by UP Cell Biological Society. This year's SIHAY zooms into the world of bioinformatics, decoding archives, unraveling the biological code through molecular data acquisition and analysis. With the advent of technological advancements that urge to unravel the closed book science, bioinformatics proves to be a principal tool in collecting the essential codes to grant access towards the bigger world of biology. As we witness the hybridization of biology with other established fields in decoding the codes of life, let us look forward to bridging the gap between science and technology in evidence-based and precision studies through bioinformatics.
SIHAY NBO is comprised of several contests and sub-events, one of which is Bio-Symposia. Bio-Symposia aims to present recent advancements in biology and biological instruction to students and educators in the hope of using this knowledge to innovate their learning and teaching styles and methods for the advancement of biological education. Last April 25, we tackled Bioinformatics for the betterment of the Philippines, as discussed by our guest speaker, Sir Carlo Lapi, a senior bioinformatics specialist at the Philippine Genome Center. Today, we will be tackling a different topic on bioinformatics with another acclaimed speaker. Are you guys ready to be bio-informed?
And just a quick recap, SIHAY 2021 started off our first Bio-Symposia seminar with our acclaimed speaker, Sir Carlo Lapi. But the week is long and was absolutely exhilarating. This was then followed by the science feature writing contest, and surely it was a close contest among our young writers last April 26. On the third day of SIHAY 2021, the creative minds of both junior and senior high school students were challenged in the digital poster making contest. On the same day, high school students were put on a much heated elimination round in the Junior Life Sciences Research Olympiad, while the wit of the students in biology were tested on our Biology Quiz Olympiad's elimination round last April 28. The final round of the Junior Life Sciences Research Olympiad was held last April 30. And certainly, the judges had a difficult time in determining the best of the best research paper. And earlier today, the final round of Biology Quiz Olympiad was conducted. The winners in every event will be published on the SIHAY National Biology Quiz Olympiad's official Facebook and Twitter page, so please stay tuned.
Before we proceed with the event proper, we would like to encourage our attendees and viewers to use our official hashtags, #Biosymposia and #BeBioinformed, in sharing your thoughts about our webinar in your individual social media accounts.
We would also like to invite our attendees and viewers to please like, share, and follow the official social media accounts of the UP Cell Biological Society on Facebook @UPCells, Twitter @UPCells, and Instagram @TheUPCells. You may also want to follow us on our SIHAY NBO Facebook and Twitter page @SIHAY NBO to keep yourselves updated on the National Biology Olympiad.
Soon we will find ourselves in a vast horizon of knowledge. So sit back, relax, listen, and prepare to learn and gain a new perspective about bioinformatics.
Before we begin, please be heedful of the following house rules. Proper etiquette is expected among all participants. Any form of discourtesy shall not be tolerated. Kindly key in your questions for the speaker in the Zoom meeting's chat box or in Facebook Live's comment section. These questions will be noted by the organization and will be addressed on our post-webinar publication. Additional questions for the speaker shall be entertained through email. And lastly, to acquire e-certificates, attendance on the two webinars are required, as two codes will be asked in the post-webinar form. The second code and the post-webinar form shall be announced at the latter part of this webinar. But of course, this seminar will not be possible without our sponsors and partners. Hence, we would like to acknowledge them for making this event possible. In cooperation with UPLB Computer Science Society,
UP League of Agricultural Biotechnology Students,
Don Honorius Ventura State University Biological Society,
Pampanga State Agricultural University Biological Society,
Biological Society Laguna State Polytechnic University Santa Cruz Campus,
PLM, Society for Biological Sciences.
Bicol University Symbiosis.
League of Biology Enthusiasts. With Media Associates, UP Society of Management and Economics Students.
UP Biological Sciences Society.
Philippine Society of Cell Biology.
Biology Program Council. Special thanks to UPLB Development Communicator Society
Symbiosis, the UPLB Biological Society.
and Agham Youth UP Diliman. Again, we would like to remind everyone of the following house rules. Proper etiquette is expected among all participants. Any form of discourtesy shall not be tolerated. Kindly key in your questions for the speaker in the Zoom meeting's chat box or in Facebook Live's comment section. These questions will be noted by the organization and will be addressed on our post-webinar publication. Additional questions for the speaker shall be entertained through email. To acquire e-certificates, attendance on the two webinars are required as two codes will be asked in the post-webinar form. The second code in the post-webinar form shall be announced at the latter part of this webinar.
We would like to encourage our attendees to use our official hashtags, #Bio-Symposia and #BeBioinformed in your social media accounts when sharing our event.
We would also like to invite our attendees and viewers to please like, share, and follow the official social media accounts of the UP Cell Biological Society on Facebook @UPCells, Twitter @UPCells, and Instagram @the UPCells. You may also want to follow us on our SIHAY NBO Facebook and Twitter page @SIHAY NBO to keep yourselves updated on the National Biology Olympiad.
To formally start our Bio-Symposia, Elijah, will you do the honors and introduce our acclaimed speaker for today?
Our speaker for today will give us his keen insights on the opportunities and challenges in bioinformatics in the Philippines. He graduated with a bachelor's degree in computer science and took his postgraduate degree in computer science at the University of the Philippines, Diliman. He also obtained his doctorate degree in computer science from the same university. Presently, he is a research faculty member and the director of the core facility for bioinformatics at the Philippine Genome Center. He is the current director of the Computational Genomics and Systems Biology program. We are pleased to welcome our esteemed guest speaker for today, Dr. John Michael C. Yap.
A little bit of a sneak peek of what this lecture will be about. Okay, so there will be more major topics. Start off with a little bit of introduction, at least to give you a view or a partial perspective of where I'm coming from. So, what is bioinformatics? Um, the most fundamental is a multidisciplinary and oftentimes an interdisciplinary discipline that analyzes biological data from different levels of granularity, from the genetic level even up to the ecological level, okay, using different perspectives, okay, like from a biochemical perspective or chemical perspective to a biological perspective, right, and using computational, mathematical, statistical, and even engineering perspective. So I've mentioned analysis or data analysis since we are in the midst of Industry 4.0, data-driven economy, right? We could also view bioinformatics as applied data science, particularly because a lot of the applications of the field entails analyzing voluminous amounts of data from usually from open databases or sometimes generated by the scientists. So that said, what are the bioinformatic things that one can do, right? Now I've mentioned data analysis, and you cannot actually work or perform any data analysis or data science without data, right? So even at the most fundamental, developing biological databases to store different types of information from different types of species and different levels of organization. It's actually, you know, a pressing need, still a pressing need for in the field of bioinformatics and in the field of the life sciences, especially when you take into context, local needs or local researches that are being done, right? There's a need for repository, okay, where researchers can store the data that they have generated or they have collected, well as for them to be able to curate, manage, and to allow access and even partial analysis of other researchers um, to their own data, right? Now, of course, that's for the data collection and data management part. Now, what kinds of analysis? We now move to the analysis part, right? What types of analysis can one do in bioinformatics? Well, for starters, you could analyze similarities between groups, excuse me, or sets of biological sequences. Okay, mostly it's usually DNA sequences or amino acid sequences, to see if certain species um, share certain similarities um, at certain positions in a biological sequence, or if between species there are certain variations that exist within their genomes or within their proteomes. That is to say, their genetic sequences or their amino acid or protein sequences. Right? Now, it's also possible that you might have a sequence data at hand, but you don't know what type of organism uh, owns or has that kind of sequence or partial sequence. So what you could do is that you could actually search databases to see which species could possibly have that kind of sequence or genetic signature, right? So you have your sequence similarity search. Basically, you have the database, right, which might contain your sequence. Then you could perform a database search just to check if there are um, species which are closely resemble, whose genetic sequence closely resembles the one that you have at hand. Now, it's a bit different than your usual information management system because here you try to maximize the similarity of the entire sequence given your query. Hence, this is actually a non-trivial um, type of analysis. The most or the de facto standard, the algorithm that is used for sequence similarity search is BLAST algorithm. So now that so those are the basic analysis that you do. Sequence similarity in effect, right? So you could analyze several sequences by aligning them or trying to search for which sequence or which organism actually has the most similar sequence given your query sequence. Now, assuming that you have the similarity at hand, okay, you could actually sort of generate a family tree um, based on the similarity, okay, in which case our sequence alignment, um, each species has on a genetic level or even on a proteomic level. Now, this is important because, or at least this is interesting to life scientists because um, here you try to check the evolutionary path that, or the evolutionary divergence of your set of species that you're trying to analyze, right? In some cases, if you um, add latent data, okay, or latent metadata associated with each species, you could even infer the approximate date at which certain evolutionary events or mutations have occurred, given your set of species, right? Okay. Now, that's usually for interspecies analysis. Okay. Within species, you could, or even within the sequence of a particular, what we call this organism, you could also try to find recurring patterns within the genetic sequence or the proteomic sequence that may um, have some functional importance or biological significance for this pattern. Here you perform what's called motif finding. So basically, you try to find the most common subsequences or portions that are recurring in a particular sequence. Okay. This might be important because it could be um, you know um, signs or evidences of conservation or of replication or crossover events. Okay. that might be of significance to uh, to infer evolutionary um, events that occurred in a particular species. Now, some another thing that you could do, right, as part of the analysis or as a bioinformatic analysis is that you could assemble sequences. Okay, what does this mean? Now, normally in a wet lab, right, when a wet a sequencing lab gives you the output of a sequencer, what you're usually given are snapshots of the entirety of a particular portion of, say, a genome or even the entire genome. Now, since you're only given a snapshot, right? Um, one of the tasks that you might want to do to be able to proceed with subsequent analysis is that you want to generate what the entire genome of your target species or organism looks in its entirety, right? And here you perform what's called sequence assembly, or generally termed as or popularly termed as genome assembly. Um, you could do it by uh, trying to piece together the sequence trees, the output of the sequencers, based on some reference sequence, assuming that there is an existing reference or gold standard genome available for your uh, what do you call this, for your species in focus, or you could do what is called de novo assembly. Basically, as opposed to a reference-based one, you try to assemble the sequence without any reference at all, in which case you're generating more or less an unbiased sequence or genome. For that. Okay. Again, these are non-trivial tasks, particularly because they take up too much memory, right, or RAM that matter, or at the get-go or in general, it takes up too much time to be able to come up with a perfect sequence assembly. So next up, once you have your sequence assembly, right, you could try to analyze um, which positions, right, are hotspots for variation or mutation. Hence, you have what is called variant calling or SNP calling. SNP stands for single nucleotide polymorphism. Okay, this is on the assumption that the hotspot is only happening at a single point or a single point locus. Okay, only one nucleotide um, or nucleotide position is deemed as a hotspot for mutations. But it is also possible that it could be a contiguous area, right? Locus. So in general, you have variant calling. You try to look for variants or places or loci within a super sequence, a long sequence, or even a genome where a significant amount of variation occurs when you align the reads that map to that particular position. So there. Now, a little bit of the usual sequence analysis. You have gene expression analysis. Excuse me. Now, we've known from basic biology, right, that um, the expression of certain traits are influenced by your genes or certain genes. The idea now, the question is, um, you now have several genes, okay, or several you call them coding regions, right? mRNA messenger RNA producing regions in your genome. Okay, which of those genes are responsible for the expression of a particular trait, right? So you perform gene expression analysis. Okay, at its most fundamental, you try to analyze, okay, um, which genes have significant differential expression, right? Okay, when you compare two sets or two groups of samples, one where a particular trait is not expressed and in another where a particular trait is expressed. And then you try to analyze which uh, which along the genome, okay, or which genes amongst the genome, okay, have significant differential or differential expression, or where the measured expression is statistically significantly different, okay, between the two groups. So in that regard, you try to identify candidate or even putative genes that are responsible for the expression of a particular trait or the inhibition for that particular trait. Okay. Of course, that's the most fundamental. Among other things, is that you could also try to provide some sort of functional, what you call infer some functional mechanism uh, based on the co-expression of certain genes subject to a particular trait. So now you try to group together genes that are co-expressed or similarly expressed um, in given a particular trait. So that's for gene expression analysis. Now, beyond that, right? Apart from getting functional similarities, uh, what they call this, between sequences and generating family trees or analyzing variations within the genome or even gene expression, one such analysis that could also perform is called structure prediction. So now the idea here is that given your sequence, right, analysis, what does it look like uh, in reality, right? So you're just given a text file, okay? Your task now is to predict what does it look like in 3D, right? That is to say, the structure. Could be secondary structure, this is ternary structure, quaternary structure. Basically, what does it look like in reality or as close to reality, right? Okay. This is very important for proteomics analysis in particular, right? And one such applications off the top of my head would be, for example, drug discovery or um, analyzing, um, the effect of a particular pathogen to its host by analyzing the protein, protein structure and how it acts or behaves based on its structure. And attached to this, you can also have what is called molecular docking. Okay, so remember when I said drug discovery or drug development for that matter. Now imagine that your drug is chemically or biochemically produced, right? Then it has to have a way, okay? And using this, or I'm illustrating this example simplistically, right? There must be a way for it to attach itself to the pathogen which it seeks to either inhibit or um, basically inactivate, right? Um, so to be able to do that, right, one uh, computational way of inferring how your uh, drug design works is by performing molecular docking simulation. It's kind of like a trivial thing. Actually, if you think about it, a lot of the analysis or the problems that bioinformatics algorithms seek to solve sound like trivial matters, but they're actually not, right? Um, the molecular docking is one of them. It's just kind of like trying to fit, right? The shape sorter problem that kids or babies usually do. But it's a little bit more complex than that, particularly because there are a lot of biochemical and biophysical considerations, forces, chemistry that you have to take into consideration to make it work. So it doesn't necessarily mean that if, for example, here you have a target and you have a ligand, and um, you know the state quote unquote of the ligand is similar to that of the target where it seeks to dock, okay, it will already work. You also have to consider the chemical composition of the ligand and the target, and of course the physical, the physics that is attached to their chemical composition. So here, for molecular docking, we try to simulate in silico, in silico, I mean, computationally, that is, uh, if uh, how you know molecular molecules fit together. Also, this is not a uh, computation speaking, this is not a trivial matter because of the physics and the chemistry involved. So you'll have to compute hundreds to thousands of equations just to come up with an accurate simulation of such, which in turn might take a couple of hours or even days to be able to perform. And then finally, uh, at least in the context of my lecture, okay, you also have biological network modeling and simulation. Now, oftentimes, for simplicity's sake, we try to analyze components and organisms or bit molecules um, by its lonesome or out of any system. But in truth, no molecule, no component, no organism works out of a system or outside of a system. So here you try to analyze biological components and phenomena in the context of a biological system. And to be able to do this, you model it. The simplest way of modeling a system is a network or a graph in this case. So you have here nodes, right, which might represent certain components. In this case, this is a gene network. So each node is a gene. And then each gene is connected to some other gene based on some mechanisms. Okay, in this case, this is a gene regulatory network. So each node here is a gene and it's a um, unidirectional node. So basically, the influence or the regulation happens one way. So for example, HAP22 here, the green one in the middle regulates SINA, WRQ23 and ASN1. And in turn, some of those nodes are um, are regulating other genes or nodes, right? And then if you notice here, not only do you see the regulation pattern, but you could also see here cluster, okay, which could also mean some sort of functional significance or biological significance, right? So for example, here you have the light blue one here is involved in energy production, particularly in glycolysis and gluconeogenesis. You have here the C metabolism, carbon metabolism, right here, the orange part, and then you have the yellow one involved in transcription, something like that. So you could analyze also clusters or groupings, right? And how each of those groups affect one another. So at least those are some of the simplest things that you can do. And you could see now here that it involves lots of data points. And as such, this is where you could also um, consider bioinformatics to some extent as big data analysis or big data science. So those are some of the things that you could do for in bioinformatics. Okay. Now, what are the bioinformatics things that you could do here in the Philippines? So now, uh, I'll just mention some of the analysis that you could do. But what I want to show here are some of the applications, the researches that are being done currently here in the Philippines. Now, for the purpose of this talk, I would just like to group uh, at least what I observed to be three major application themes of bioinformatics in the Philippines. Of course, the lowest hanging fruit here is health application, right? So, in particular, human health applications. Excuse me. Here, some of the active participants in bioinformatics for health applications are the UP National Institute of Health. You have the DOH Research Institute for Tropical Medicine, um, and also, of course, CDC, right? Okay, so what are the health applications that I am talking about here, right? Well, you could actually do, in a sense, disease profiling on the genetic level. So you try here to identify, for example, genetic determinants or indicators of certain diseases, especially the ones that are heavily affected by heredity, right? Like in this case, in biotic, we have a type media, but you also could also have visual suspects like cardiovascular diseases, diabetes, right? And other lung and also cancer. So you try to identify genetic determinants, that is to say, what makes a person susceptible to certain diseases or syndromes. Now, on the one end, you could also profile the pathogen itself, right? Like, for example, I think the hot topic right now is, of course, COVID. And one of the things that we've done is, to analyze certain SARS-CoV-2 um, samples that we have, and from there we actually were able to identify which variants are what they call this are present in the Philippines, and we were even, I don't know, from an academic standpoint, fortunate. From a public health standpoint, unfortunate that to identify an emerging SARS-CoV-2 variant from samples derived here in the Philippines, the so-called Q3 variant, right? And it's not only that. For example, you could also check other pathogens like, for example, for tuberculosis, HIV, even the common flu, to be able to understand the behavior, the mechanisms, and you know the um, the uh, transmissibility of certain diseases, of certain pathogens, which in turn would um, uh, what they call this cause a particular disease or syndrome. Now, uh, one other application, and I think this is close to UPLD's heart, is it's agricultural applications. For agriculture, I do not only mean crop plants. Of course, rice comes into mind. You have dairy, right, which is uh, technically in LB, the LB campus, right? You also have field rice, which is in Nueva Ecija, but you also have applications which cover livestock and fisheries. Like, for example, you have the Philippine Carabao Center, and you also have the Fishery Bureau. I think I forgot the name. Really sorry about that. But the idea is, um, you try to for agricultural applications. Of course, the idea there is to come up with a way to increase yield of crops, right? Uh, or improve the reproductiveness and the quality of meat or output of a particular livestock animal, right? And at the same time, you could also analyze how to protect them from certain diseases themselves. So there's an agricultural health application, like, for example, for um, for rice or for crops, you have the blight and other bacterial infections. Similarly with the animals, like, for example, one of the hot topics right now is African swine flu. You try to analyze, or do biosurveillance of if certain pathogens actually exist or is present locally, right? And also you try to analyze uh, certain um, characteristics of uh, crops or animals, right, or livestock that would have them be immune, right, for to certain biotic or biotic stresses, right, and even possibly develop therapeutics or mitigating measures to be able to counteract those effects. And then, so that's for agricultural applications. Now, finally, we also have biodiversity applications, right? So we know that the Philippines is a biodiversity hotspot, and as such, there are a lot of flora and fauna that are present here and even have yet to be discovered. And here, genomics and bioinformatics can greatly help analyze the extent of the diversity, as well as to identify the species. Okay, at least that could complement taxonomic classification at the you know using physical characteristics. So, off the top of my head, um, I know that the UP Marine Science Institute and the UP Institute of Biology have been doing biodiversity based bioinformatics application, right? So here they try to characterize, right, and to even quantify the diversity, the level of the genetics of the flora and fauna, and even to identify new species, right, that have otherwise that are otherwise genetically significantly genetically different, right? Um, Even if there seems to be no physical uh, characteristics that are different from existing known or documented. So those are some of the uh, applications of bioinformatics, right, that are being done and can be done in the country. Of course, there are many others, but I would like to focus on these three because these are the lowest hanging fruits, or at least the ones that where you could use as a jump point. Now, with the opportunities, come the challenges. Okay, so what are the things that at least I would consider some sort of roadblock when you try to make a bioinformatics career or try to engage in bioinformatics here in the country, right? So I present here some of the things that at least I have experienced or have observed over the past couple of years I've been in the field, right? So of course, one of the things here is that availability of data, not necessarily data per se, but data, you know, um, in the local context. Those are that are sourced in the Philippines. Um, uh, Not saying that the local scientists are greedy, but sometimes um, making them available or publicly shareable is still a problem in the Philippines. Okay, of course you understand the intellectual property, but at least in the interest of open science, to be able to make any field flourish, right? It would be nice if there would be some openness, but in the terms of sharing the data, right? Of course, it's also a problem of what platform are they going to use, right? We have, of course, existing ones like NCBI, DDBJ, kind of thing. But it gets lost or it gets mixed up with other data that come from foreign countries. So sometimes it's tough to really recognize that. So maybe a local repository would greatly help in making sure that, hey, we have data here in the Philippines, and you could use basically a shout out to all the other budding research that there's existing data that comes here from the Philippines, and we could use that. Then, of course, there's also the problem of generating the data itself, right? So it's not exactly cheap, right, to generate genetic sequences, and that's also a problem of economics. Okay. Of course, there are other ethical considerations that you have to um, consider. Of course, the lowest hanging fruit would be for health applications. You'll have to pass through usual bioethical processes to make sure that you're not violating any uh, you know rights to privacy or unwarranted right to information. Right? But there are also some other things that you have to uh, consider. Like, for example, in the context of agriculture, if there's an outbreak, right? If you a part of the information that you might have to you will definitely need is location data. But if you make it too specific, so much so that you could pinpoint who exactly, which farm or which resident did the infection emanate. Okay, then that could be if not a breach of privacy, it could be you know a source of problem in terms of possible invitation to discriminate certain individuals, right? Just because they were the source of infection or the source of a particular pathogen. So those kinds of things. The ethical considerations here not only not only is limited to um, biological implication, but also to social implications. The next one would be human resource capacity. Uh, yeah, I guess that's the reason why I'm also open to these kinds of invites to, uh, to help, I suppose, entice new generations of bioinformaticians or of potential bioinformaticians, especially from your lab. For the most part, PGC's mandate is also to capacitate um, researchers to be able to do genomics and bioinformatics research, or at least analysis, right? But um, we are also what's it called this? We are also aware that not it's not sufficient. Okay? Um, that we hope that those that we have trained could also train other people so that there could be a multiplier effect. Um, but the fact remains, human resource capacity is also still a challenge in Philippine science. Uh, then we also have physical resource capacity. Like I've said, a lot of the bioinformatics analysis uh, entails um, loads of computational resources. And of course, with the advent of cloud computing, people could avail of that as well. But sometimes it's either unaffordable or because of the way cloud uh, services are structured, it's not um, feasible from a if you take into account data privacy or make sure that sorry when you try to analyze data that has not yet been published or are not that fit for publication, so you still have to have some sort of protection for your intellectual property. In that regard, as much as possible, you want to rely on uh, computational resources that are within country. uh, Unfortunately, it's not yet as ubiquitous, right? um, there's still a lot additional blood needed, especially in um, outside Metro Manila, or at least the usual science hubs, right? If you want to make bioinformatics or science in general inclusive or accessible to other people, then there must be um, um, at least a substantial um, provision of such computational resources that are accessible to areas that are otherwise distant from. There you go. Next is sustainability, right? Uh, a lot of the bioinformatics, probably at least outside academia, right, are a lot of the endeavors in bioinformatics are largely project based. So what does that mean? The the human resources and the physical resources that are allotted for such bioinformatics are only as long as the project is still being implemented. But if the termination project, the project has already terminated, it has already reached its end period, right? And no additional funding is being given, then it's being allotted elsewhere. And the human resources are also human resources, the people, the bioinformaticians are not being held on to, and they venture out into other fields as well, or at the very least, they get opportunities outside the Philippines, which is kind of sad because we already have a human resource capacity problem, and you still are being subjected to brain drain. So in a sense, there must be some way to make this what you call the sustainable, um, especially for retaining human resources or bioinformaticians here in the country. And speaking of sustainability, attached to this one is that, like I said, it's mostly academia and public sector focused. So the bioinformatics researches or endeavors are usually projects, right, that are being conducted by universities and or the government. There is still no mainstream local industry that has bioinformatics, save for a very very few um, hospitals, okay, as I would say, right? But it's not as prevalent, especially since we've seen how, we're industry. I mentioned we're in the industry 4.0. We're in the data-driven economy, and yet uh, there's still no place in the industry for bioinformatics, at least a locally and um, and widely. Yeah. So these are some of the challenges I would say that are faced by those who would like to choose informatics here in the Philippines. And with that, I end this presentation. So thank you very much.
Thank you for that very informative lecture, Dr. Yap. It is indeed inevitable to different fields of science that there would still be challenges that may arise no matter how advanced these fields are. Yet, even with only the current local resources they have, they still persevere in acquiring the necessary information that are vital to the establishment and development of the field of bioinformatics. It truly has a wide range of data that are deemed helpful in various studies and researches.
Totoy and Diane, again, thank you for that very insightful discussion, Dr. Yap. Bioinformatics is not only subjected to biology and computer science, but also encompasses the fields of mathematics, statistics, chemistry, biochemistry, and engineering. It surely does sound like a very interesting field of study.
We are now beginning the open forum on the discussion of Dr. Yap. For our Facebook Live attendees, relay your questions in the comments section. Your questions will be addressed in our post-webinar publication or through email.
While waiting for the question to come through, I just want to express my sentiments regarding the discussion of Dr. Yap, where apart from the analysis and prediction of gene and protein structure and function, gene variation, gene expression, bioinformatics in the Philippines is being utilized for a vast array of vital applications, such as genome sequencing and analysis of different variants of viruses, including SARS-CoV-2. The country also takes advantage of bioinformatics to cultivate agricultural lands and analyze and preserve the country's biodiversity.
I agree with you, Elijah. Bioinformatics undoubtedly serves to be a very critical field in our current situation, which is also why, even with the unfortunate situation we are in, we, the UP Cell Biological Society, are conducting these webinars to enlighten everyone about bioinformatics and its role in our present circumstances. How about our audience? Do you have any comments about the discussion of Dr. Yap? Want to share sentiments about bioinformatics? Share it with us through your social media accounts using our official hashtags, #Bio-Symposia and #BeBioinformed.
So a question from Raymond R.V. Lyrit po. In what circumstances is it more appropriate to use de novo assembly or mapping for sequence assembly?
De novo assembly is uh, appropriate if you don't have a um, a reference sequence available, or if you want, to a certain extent, if you want to, even if there is, if you want to somehow validate, uh, you know, a reference-based assembly that you did, then you could also do de novo assembly as well.
For the second question po, it's from Armel Pequeña po. What does the result of a molecular docking study imply regarding an anti-disease drug design objective?
Okay. At the very least, it gives the wet lab designers some preliminary data, and to a certain extent, a good hypothesis regarding the validity of their drug design. In a sense, okay, I guess I need to also impress this on the people, right? There are often times that they readily um, um, use the output of any analysis, not only in bioinformatics, no, but any decision support tool, okay, software application as the ground truth. That is not, while that may be convenient, that is actually a little bit dangerous. You should still somehow validate it with existing, information, literature, or studies before imputing the validity or the truth of the output of the analysis. Bioinformatics is by and large a decision support system or a decision support field. It gives you a fast way to come up with a strong hypothesis or a strong inference regarding a problem posed, okay, in a biological field. That said, okay, it should help guide researchers, or especially the wet lab or the field researchers, on how to proceed with their study, or at least how to proceed with their subsequent experiments and validations.
There's a follow-up question po, Dr. Yap. And if there are free softwares for student researchers in the Philippines to use in their methodology?
There are free softwares? Definitely there are. In fact, the core facility for bioinformatics espouses the use of free and open source software whenever possible, because you know it's also in line with our advocacy of open science. So open source technologies help, especially the free ones, not only help with the economic considerations of conducting the course, it also allows the researchers, or us in particular, to tinker a little bit with the tool, make sure that um, it is working according to what it is advertised to do. And at the same time, it also allows us some customization to be able to make the application work in accordance to our objectives. So yes, definitely, you could actually Google a lot.
Thank you, Dr. Yap.
For the next question po, it's from Joshua Maravilla. With the challenges presented, what are your advices to starting researchers if they want to conduct studies involving bioinformatic techniques?
Okay. If you're coming, okay, from a technical aspect, okay, if you're coming from the computational mathematical physical sciences, um, you may want to brush up on your genetics, on your biology, and also to get in touch with researchers or faculty members from the life sciences, right? ultimately treat it as a some sort of application field for your computational slash mathematics slash statistical knowledge. Now, if you're coming from the biological or life sciences, brush up on your mathematics, uh, particularly on your algebra, if you can, and a little bit of your calculus, right? And or at least and of course your mathematical logic. And if you could practice a little bit of tinkering with Linux, Unix-based system, could be Mac OS, could be any different flavors of Linux, and work with the command line um, interface, that would be a good start. Of course, as with my advice with those coming from the computational sciences, get in touch, man, with mathematical, statistical, or computational researchers and pass the numbers. Remember that by and large, this is a multidisciplinary field. So as much as possible, interaction with those from across the other end of the spectrum is vital. There's still, interaction with those from across the other end of the spectrum is vital. There's still, more or less, there's still a social aspect to the concept of bioinformatics. Okay. And now that's for the technical part. For the foundational part, I guess if you really want to, what do you call this, um, stay or basically, uh, have some longevity in bioinformatics, right? Find something that you think is worthwhile to solve from a bioinformatics stand. At least, beyond the technical aspects, your motivation is key to be able for you to have that drive to um, stay in the field and to flourish and to study even things that are usually beyond your current skill set, right? If you find something that motivates you, find a cancer, find a way to make sure that the crops are better yield than what it is now. Make sure that the biodiversity in the Philippines is preserved. Uh, you will be surprised on how far that will take you, and it'll motivate you to even enrich your technical knowledge. Um, there you go.
Thank you, sir.
So for our next question po, from Atina Lopango, what are the possible paths, graduate or postgrad opportunities that we can take if we are to pursue bioinformatics research career that are available in the Philippines?
Okay, here's the tricky part here in the Philippines. There's not, at least to my mind, well, there's no bioinformatics, at least, bioinformatics strictly degree, postgraduate, graduate degree yet. Although, if you want something very close to that, at least something that you could earn by degree, I believe the University of the Philippines Manila has bioinformatics as a track under their health informatics graduate program. But you'll still be focused more on health informatics in that regard. However, um, and this is going to be a little bit biased, right? If you really want to focus on the techniques, right, you could go into a computer science um, related graduate or postgraduate program, right? Um, but uh, but there are also certain life sciences uh, programs that nurture or that allow for some sort of applied bioinformatics specialization, like for example, in the surprisingly in the Marine Science Institute. Yeah. Although you're getting a marine science degree, because of the extent at which bioinformatics is being applied in a lot of their courses, particularly in the specialization, then that counts for something like a formal bioinformatics training graduate level. course, you also have the molecular biology and biotechnology. In B, I believe there's the genetics, plant genetics, yeah, plant genetics program. Um, and under the Institute for Plant Breeding, and also in the breeding, and also in the biotechnology program as well. So also the computer science. So you know, at least here in the Philippines, those are the things. So those are what I could remember off the top of my head that offers some sort of formal bioinformatics training or course. And also in the biotechnology program as well. So also the computer science. So you know, computer science. So you know, at least here in the Philippines, those are the things. So those are what I could remember off the top of my head that offers some sort of formal bioinformatics training or course.
Thank you po, Dr. Yap. Due to time constraint, we will be entertaining three more questions before concluding. Thank you.
The next question po is from Iza Thurui Pagano. In relation to medical science, how has bioinformatics influenced the application of gene therapy?
I'll be honest, I'm not sure how if we're going to strictly go with the question of how has bioinformatics influenced the application of gene therapy. But the top of my head, bioinformatics can definitely be used in the application of gene therapy. Like I said, similarly in the context of doing molecular docking simulations for drug design and drug discovery. So, for example, in gene therapy, also the computer science. So, you know, at least here in the Philippines, those are the things. So those are what I could remember off the top of my head that offers some sort of formal bioinformatics training or course.
Thank you po, Dr. Yap. Due to time constraint, we will be entertaining three more questions before concluding. Thank you.
The next question po is from Iza Thurui Pagano. In relation to medical science, how has bioinformatics influenced the application of gene therapy?
I'll be honest, I'm not sure how if we're going to strictly go with the question of how has bioinformatics influenced the application of gene therapy. But the top of my head, bioinformatics can definitely be used in the application of gene therapy. Like I said, similarly in the context of doing molecular docking simulations for drug design and drug discovery. So, for example, in gene therapy, you could do simulations, of course, right? You could do a sequence analysis at the most fundamental to see possible compatibility issues. I'm using this in a very, very loose term. And like I said, to do simulations that would otherwise give confidence to the application researcher, whether to proceed with a particular therapy or if there should be other things to consider for that matter.
For the next question from Paula Nerpio, how can bioinformatics contribute to the development of forensic science in the Philippines?
Well, definitely, it could speed up a lot of things, right? Particularly in information management alone, right? And also in the analysis of sequences. At least in terms of genetic or genomic forensic science or the genomics application of forensic science, bioinformatics can definitely help there by providing computational tools to speed up the analysis, particularly, for example, in looking for similarities, for example, in the DNA for example, a criminal or a missing person with an existing database. In terms of information analysis, it's huge. Also, in the development of other forensic methods of ascertaining or what do you call this? Uh, Ascertaining or inferring identities, to be example. However, there's also a caveat there, right? As I've mentioned, one of the things that you have to consider as part of the challenge is the ethical consideration, right? You also have to wrestle with data privacy issues, right? Not only data privacy, rights to privacy, right? Because remember that at least for humans, especially for humans, right? Your DNA, right, or genetic sequence also contains partial information regarding the rest of your family, or your ancestry, or even your descendancy. So those are things that you also need to take into consideration. Remember that in science, it doesn't mean that if you can, you should, right? You also have to uh, look at the bigger picture and take into account the social, even possibly political um, aspects or issues that might crop up when you engage in a particular type of research or experiment.
Thank you po, Dr. Yak.
And for the last question po, it's from Alea Marie Simbulan. The question po is, what is the current status of bioinformatics in terms of exploring bacteriophage resistance?
Okay, I'm not really sure of the exact state, right? Nor am I going to pretend to be knowledgeable about it, right? However, I had the fortunate, um, what they call this, activities in the past couple of weeks of interviewing various, researchers or various scientists, right? And a lot of them mentioned, having data various scientists, right? And a lot of them mentioned, having data on bacteriophages or bacteriophage data for that matter. So at least in the Philippines, there is a healthy ecosphere, I suppose is the term, for that research. There is a healthy ecosphere, I suppose is the term, for that research. In terms of bioinformatics, right? They've mentioned that the the fundamental problem of having a repository that would house locally sourced data uh, is there. Okay, and modesty aside, it's actually one of the things that I've mentioned the project. One of the things that we're trying to um, resolve is to develop a local repository, a locally housed, nationally housed repository for viruses, virus-like, and viroids, like viroid sequences. Hopefully for not only for, of course, for therapeutics and diagnostics, but also for scientific and academic research. So that's one. Of course, the analysis is um, the analysis, particularly in the characterization of the bacteriophages, is also in play. They've mentioned that they've been doing very basic sequence analysis, which of course in turn um, includes bioinformatics and it's part of the field. The thing. Not sure about bacteriophage resistance though, but at that much and it's that much.
Thank you very much, sir.
Ayan, it looks like we are running out of time. For other queries from our audience, kindly key in your questions on Facebook Live's comment section so that we can compile and send these queries to our speakers. Answers will be posted in our post-webinar publication. Now, moving on, We would like to take this opportunity to formally extend our gratitude to Dr. Yap for giving his time and effort in sharing his knowledge for the purpose of this seminar. The UP Cell Biological Society presents this certificate to Dr. Jan. We would like to take this opportunity to formally extend our gratitude to Dr. Yap for giving his time and effort in sharing his knowledge for the purpose of this seminar. The UP Cell Biological Society presents this certificate to Dr. John Michael C. Yap. Allow me to read the citations. This certificate of appreciation is presented to Mr. John Michael Yap, PhD, for his invaluable contribution as a speaker in Bio-Symposia Advanced Bioinformatics for research purposes as part of SIHAY National Biology Olympiad 2021. Given this first day of May 2021, signed by the President of the UP Cell Biological Society, Mr. Bruce John S. Estillo, and the Educational Committee Head, Ms. Princess Amy R. Deliosa.
I am sure that everyone's curiosities were piqued.
I agree, Diane. This seminar is surely in line with this year's SIHAY National Biology Olympiad. SIHAY NBO is a national inter-high school biology congress organized by UP Cell Biological Society. This year, SIHAY zooms into the world of bioinformatics, decoding archives, unraveling the biological code through molecular data acquisition and analysis. With the advent of technological advancements that urge to unravel the closed book science, bioinformatics proved to be a principal tool in collecting the essential codes to grant access towards the bigger world of biology. As we witness the hybridization of biology with other established fields in decoding the codes of life, let us look forward to bridging the gap between science and technology in evidence-based and precision studies through bioinformatics.
SIHAY NBO is comprised of several contests and sub-events, one of which is Bio-Symposia. Bio-Symposia aims to present recent advancements in biology and biological instruction to students and educators in the hope of using this knowledge to innovate their learning and teaching styles and methods for the advancement of biological education. Last April 25, we tackled Bioinformatics for the betterment of the Philippines, as discussed by our guest speaker, Sir Carlo Lapi, a senior bioinformatics specialist at the Philippine Genome Center. Today, we discussed with another acclaimed speaker, Dr. John Michael Yap, a research faculty member, director of the Core Facility for Bioinformatics and Computational Genomics System Biology Program at the Philippine Genome Center. Isn't that awe-inspiring? Got anything to share about the informative discussion of Dr. Yap? Or anything that you would like to tell your friends and relatives about this webinar? Go ahead and use our official hashtags, #Biosymposia and #BeBioinformed. Let us spread the word to everyone about the intriguing world of bioinformatics.
What a constructive discussion bestowed to us by Dr. Yap, no? I feel like I want to share what I learned in this webinar.
Me too, Elijah. Tara, tweet tayo natutunan natin, like, parang gusto ko na atang mag-cell and molecular biology major. #Biosymposia, #BeBioinformed. Ganon.
Ganon. Sige, habang nag-tweet ka dyan, Diane, we would also like to invite our attendees and viewers to please like, share, and follow the official social media accounts of the UP Cell Biological Society on Facebook @UPCells, Twitter @UPCells, and Instagram @TheUPCells. You may also want to follow us on our SIHAY NBO Facebook and Twitter page @SIHAY NBO to keep yourselves updated on the National Biology Olympiad. Again, we would like to thank our sponsors and partners for supporting us in launching this event. In cooperation with UPLB Computer Science Society,
UP League of Agricultural Biotechnology Students,
Don Honorio Ventura State University Biological Society,
Pampanga State Agricultural University Biological Society,
Biological Society Laguna State Polytechnic University Santa Cruz Campus,
PLM Society for Biological Sciences,
Bicol University Symbiosis,
League of Biology Enthusiasts, with Media Associates, UP Society of Management and Economics Students,
UP Biological Sciences Society,
Philippine Society of Cell Biology,
Biology Program Council. And special thanks to UPLB Development Communicators Society,
Symbiosis, the UPLB Biological Society,
Agham Youth UP Diliman. Once again, today's webinar was remarkable, don't you agree? This has been such a productive day for us to learn about bioinformatics.
Exactly. With all the educational discourse on bioinformatics courteously shared to us by our guest speakers, it has undeniably been an enlightening week. We didn't only learn about bioinformatics, but we were also granted the opportunity to see the world of bioinformatics figuratively and literally, with Dr. Yap graciously giving us a sneak peek of his workstation in his office.
Now that we have established a background in bioinformatics, may this be the beginning of an exciting and scientific journey. As the science of the future, I'm sure that bioinformatics can lead to different breakthroughs in medicine, agriculture, and microbial genome applications.
Siyempre, hashtag Bio-Symposia, hashtag BeBioinformed through hashtag SIHAYPlug. For our attendees to obtain your e-certificates, please visit the website link shown on your screens and enter the second code along with the first code that was announced during the first webinar. The second code is SIHAY 2021, no space with letters S, N, B, and O in uppercase letters. Again, the code is SIHAY 2021, no space with capitalized S, N, B, and O as shown on your screen. Participants shall input the two codes to receive their e-certificates.
I hope that our attendees and viewers learned a lot from the discussions confirmed by our esteemed speakers of our webinars in bioinformatics.
We'd like to thank everyone for joining us, the UP Cell Biology Society, on our Bio-Symposia 2021.
Again, I am Elijah Rivera.
And I'm Diane Chongko.
And this has been Bio-Symposia 2021, Advanced Bioinformatics for Research Purposes. We hope everyone stays safe and healthy.